Hi everyone. Today I'm going to talk about cancer therapy. So, in this topic, we are going to discuss about the treatments given to the cancer patients. So, first we should know what are the goals of cancer therapy, why we are giving these treatments to the cancer patients. The first thing, we have to eliminate the cancer cells. If we don't eliminate the cancer cells, what will happen? The cancer cell will spread throughout the body, and this condition is called as metastasis. So, to avoid that, we should eliminate the cancer cells. And the second thing, we should prevent recurrence and metastasis. As I already explained what is metastasis. We know recurrence means after giving the treatment that we cannot completely eradicate cancer cells. So there is some possibility of recurrence of cancer cells. So again, the patient will get cancer tumor growth. This is called recurrence. So we need to prevent that. We have to give the proper treatment. And the third important point is it should reduce the symptoms. That is, the cancer patient will have too much of pain as well as they will have numerous symptoms, right? So we have to reduce the symptoms, and we should improve the quality of life. They have to get their normal life. So for all this purpose, we have to give treatment to the cancer patients. So these are the different types of major types of cancer therapy. So I'm going to explain one by one. The first one, surgery. So in surgery condition, what doctors will do, they will remove the tumor. It is the physical removal of the tumor. So it is very effective for the solid tumors. We cannot give this treatment to the blood tumor. It is a liquid, right? So we cannot do surgery in that case. But for solid tumors, it is most effective. And the surgeons will remove the solid tumors. At the same time, they also remove the nearby lymph nodes. Why they are doing this? Because through these lymph nodes, the cancer cells will spread throughout the body. To avoid that, the doctors, what they will do, they will remove the nearby lymph nodes also. So, while giving the surgery treatment, what they will do, they will give chemotherapy or radiotherapy. So, mostly they will give the combination of treatments, right? So, I'll explain in the next slide while explaining the chemotherapy or radiotherapy. So, in this picture, you can clearly see like the surgeons removed the cancer tumor. In the second picture, you can see the cancer tumor removed by using the surgery process, right? So when we are talking about the cancer therapy, these are the two important terms we should know. That is adjuvant therapy and neoadjuvant therapy. So what is the meaning of neoadjuvant? Means, okay, here you can see I have given the explanation. Neoadjuvant means before main treatment. Adjuvant means after main treatment. So before starting the main treatment, the doctor will give any other treatment. Why? Say here I have given example. So this neoadjuvant therapy given before the main treatment. Usually it will be surgery. So main treatment is surgery. So before surgery, what they will do? They will give chemotherapy or radiation therapy. What is the purpose of doing that? Because they want to shrink the tumor. So if the tumor size is reduced, it is easy for the surgeons to remove it, right? It is easy for the surgery process. That's the main reason. Before starting the main treatment, they are giving the neoadjuvant therapy. Any kind of therapy given before the main treatment, it is called neoadjuvant therapy, right? It may be chemotherapy, it may be radiation therapy, it may be hormone therapy, it may be any kind of therapy. In the same way, the another one important term is that is adjuvant therapy. So this therapy will be given after the main treatment. For example, uh, the surgery was over. After that, in some cases, doctors will give chemotherapy or hormone therapy or radiation therapy. So why they are giving this therapy after the surgery? So after removing the cancer tumor, we cannot tell the cancer cells are completely eradicated. No, some leftover cancer cells will be available in that place. So we have to kill the leftover cancer cells. Why we have to kill it? To reduce the recurrence of the cancer, right? So for that purpose, a doctor will give adjuvant therapy. That means the therapy given after the main treatment, right? So here, these are the examples: chemotherapy, hormone therapy, or radiation therapy. So chemotherapy, chemo means chemical. So from the name itself, you can understand here we are going to use the drugs to kill the cancer cells. So here they are using anti-cancer drugs to destroy the cancer cell. So these drugs particularly targets the rapidly dividing cells because we know cancer cells have the ability to divide very rapidly. So it can be used alone or we can use the chemotherapy along with the surgery or radiotherapy. So here we can use the different routes like oral or intravenous routes. These are the some anti-cancer drugs examples like cyclophosphamide, cisplatin, doxorubicin, paclitaxel, methotrexate. These are some anti-cancer drugs. So how they are working, we need to understand the mechanism of these drugs. So this is the core principle. These drugs mainly target the rapidly dividing cells. So what they will do with the rapidly dividing cells, they will interfere DNA synthesis. We know during the cell division, these are the important process will occur in the cell. What are the process? The DNA synthesis, mitosis, and nucleotide metabolism, right? So our drug will interfere with this process and it can able to easily stop the cell division process. So if the cell division is not occurring means what will happen? The cancer cells cannot grow further. So the cancer growth will be stopped. So in these chemotherapeutic agents, we have different classes of drugs and they have different mechanisms also. So here I have given few examples. The first one is alkylating agents. The example drug is cyclophosphamide. So what these alkylating agents will do, from the name you can understand, it will do alkylation. So by this alkylation process, what it will do, it form cross links between the DNA. So if any changes in the DNA structure, what will happen? The replication process will be blocked. The further transcription will be stopped. So cell division will not occur. This is the first drug. The second one, anti-metabolites. So what it will do, it inhibit purine and pyrimidine synthesis. This purine and pyrimidine is important for the synthesis of DNA. So it can able to prevent the DNA and RNA formation. So cell division will be stopped. The third one, topoisomerase inhibitors. So topoisomerase, we know it is an important enzyme involved in a DNA replication process. So these are the some example drugs, doxorubicin. So these are the topoisomerase inhibitors. So by inhibiting this topoisomerase enzyme, what will happen? It causes DNA strand breaks during the replication process. So DNA strand will be breaked. The replication process will be stopped. The final one, mitotic inhibitors. So it inhibit the mitosis process. That's all. It disrupt the microtubules. So the metaphase during the cell division will be stopped. So this is the picture. Here you can see alkylating agent. It induces the cross links in DNA. Antimetabolites, they inhibit nucleotide synthesis. Like topoisomerase inhibit the topoisomerase enzyme. Mitotic inhibitors block the mitosis process. So antimetabolites, it inhibit the nucleotide synthesis, purine pyrimidine synthesis. So this is the major mechanism. These chemotherapeutic agents, they inhibit, they only target the rapidly dividing cells, but it have many side effects. Why it have side effects? Because it mainly target the rapidly dividing cells, right? But naturally, our body have many different types of natural rapidly dividing cells. For example, bone marrow, so gastrointestinal epithelium, hair follicles. These are some examples for the rapidly dividing cells. They naturally in our body, very active cells, they divide very rapidly. So while targeting the cancer cells, so what these drugs will do, these drugs also affect the function of these cells. So by affecting these cells, it produces a lot of side effects like hair loss, nausea, vomiting, fatigue, mouth sores, anemia, infections, and easy bleeding. So in many people during the surgery process, they voluntarily they will remove their hair because after the treatment it will happen. So hair loss, too much of hair loss will happen. So the surgeon advised the cancer patients to remove the hair completely. So the next one, radiation therapy. So here we are using high energy radiation to destroy the cancer cells. So what radiation will do? It damages the DNA. So cell division will be stopped, cell growth will be stopped. So there are two types of radiation, external beam radiation and internal radiation. So what are the major uses? It will cure the cancer, it reduce the tumor size, and also it helps to reduce the symptoms. So as I said already, it always combined with the surgery or chemotherapy. So here, this is the first type, external beam radiation. So in the external beam radiation therapy, radiation will be delivered from a machine outside the body. Here, radiation will be given by using the machine. So the radiation will be given outside the body. This machine will not touch the patient, and the radiation we can give from the multiple direction by using this machine. It is the local treatment. For example, if the patient is having, see the picture, if the patient is having lung cancer, the radiation will be given only to the chest region. So it is a local treatment. The radiation will be given to the specific area. The second type, internal radiation therapy. So here the radiation source will be placed inside the patient's body. So it is also called brachytherapy. That radiation source will be a solid source. It may come in the form of seeds or capsules. These seeds or capsules will be placed in or near the tumor. But we have to keep this capsule inside the patient's body, right? So it deliver radiation over a period of time. So it is also a local treatment. As I said, if the patient is having lung cancer, the capsule will be placed near the lung region. So the next one is systemic radiation. So here the source of radiation is liquid source. So it will be given by oral intake or IV. So this liquid source of radiation will be injected and through the bloodstream. So our body fluid may emit radiation temporarily, only for a particular time. This particular radiation source will emit the radiation. So this is systemic radiation because the whole body will get the radiation. The next type is targeted therapy. So here it is having different types. The first one is a tyrosine kinase receptor inhibitor. So what it will do? Here mainly we are targeting the cancer cells. As I said, some normal cells also getting affected, right? So here we are if we are using this targeted therapy, the side effects will be fewer. So that is the major use of this therapy. The first type is tyrosine kinase receptor inhibitor. Here, this tyrosine kinase, see this tyrosine kinase is a cell signal for the cell division process. So we need to block this signal. So we have to stop the tyrosine kinase receptor. So we have to block the tyrosine. This is the receptor. Just imagine this is the receptor of tyrosine kinase. So if we block this receptor, what will happen? Tyrosine kinase cannot bind with the receptor and it cannot send the signal. So cell division can be stopped. In the same way, angiogenesis inhibitors, we know angiogenesis is a process of a blood vessel formation. So most of the cancer cells will form the blood vessels to get proper nutrients and oxygen so that they can grow. Um, they can get the nutrient so that they can grow very fastly, right? So we have to inhibit the blood vessel formation. If we stop the blood vessel formation, what will happen? The cancer cells cannot get the proper nutrient, so the growth will be stopped. So by blocking some particular receptors which are responsible for the angiogenesis process, we can stop this blood vessel formation. So this is the example drug. In the same way, here I have given some other examples also. So these are the advantages of this therapy. There is reduced toxicity to normal cells because we are mainly targeting the cancer cells because we are selectively targeting the particular receptor which is involved in the cancer cell formation, right? So the normal cells will be uh, escaped, right? So the toxicity will be reduced and the fewer side effects. The next one, immunotherapy. So here we are not going to use any external source. Here we are going to boost our immune system to fight against the cancer cells. So here we are using the body's own immune system to fight the cancer cells. So we are enhancing or restoring immune response against cancer cells. These agents do not directly block the growth signals, but it activate. The only thing is what we are giving the drug, it will activate our immune system. That's all. Example drug, some monoclonal antibodies. Okay. So these monoclonal antibodies will recognize the specific targets. For example, the PD-1 inhibitor drug. This removes the brake on immune cells, allowing T cells to attack cancer. So what this drug will do, it remove it inhibit this BD1 receptor. This BD1 receptor, what it will do, the cancer cell will bind with this PD-1 receptor and it stop the T cell activation. But our drug, what it will do, it block the PD-1 receptor, so the cancer cell cannot bind. Our T cell will be in active condition. It will fight against the cancer cell. Here, our drug did not fight with the cancer cell. It activate our T cell, so the T cell can easily fight against the cancer cell. So in the cancer immunotherapy, we can boost the existing immune response, or we can trigger the new immune response, or we can add the cancer fighting immune components to the body. Next one, hormone therapy. So this cancer treatment slows or stop the growth of hormone dependent cancers. Is in some cancers, the hormones are the reason for the development of cancer. So in that condition, what it will do, it will stop or slow the process of hormones. So what it will do, these drugs will block the hormone production or interfere with the hormone action at the receptors. So it is effective only in hormone receptor positive cancers. For example, breast cancer and prostate cancer. So in these cancers, hormones plays important role. So what our drug will do, this our drug will bind with the hormone receptor so that the hormones cannot bind with the receptor and it cannot stimulate the cancer cell growth. Here you can see this is the estrogen receptor. If the estrogen bind with the receptor, what will happen? It will stimulate the cancer cell growth. So here we are giving the drug. The drug will bind with the estrogen receptor. So estrogen cannot bind with the receptor. So the cancer cell will be, the cancer cell cannot grow. So this is another one important therapy, very interesting one. This is photodynamic therapy. So photo means light. So here we are going to use light activated drug. These drugs are called as photosensitizer. So they will be activated by using a specific light to kill the cancer cells. So here you can see, so person with cancer receives a drug called a photosensitizer. So the first step is we give the photosensitizer drug to the cancer patient. So in 24 to 78 hours, these cancer cells absorb the photosensitizer. So all the cancer cells it will reach because they will add some target to this sensitizer. So it specifically reaches the cancer cells. After that, the cancer cells that after absorb the photosensitizer, they are exposed to the light. Okay, so once the um, photosensitizer gets light, what will happen? It will be activated and it produce reactive oxygen species. So this reactive oxygen species will can able to kill the cancer cells, right? So here this is the example drug. This drug, when exposed to a specific laser light at particular wavelength, they have some specific rules, sorry, specific conditions. These lights, specific wavelength we need to give. So it will produce the oxygen radicals, and these oxygen radicals will destroy the cancer cells. So this is called a photodynamic therapy. Then this one, the stem cell or bone marrow transplant. So here, in this treatment, healthy stem cells are given to replace damaged bone marrow after high dose chemotherapy or radiation. So after giving the radiation or chemotherapy, what will happen? Our bone marrow will be damaged. So in this treatment, a healthy stem cells will be injected to replace the damaged bone marrow. So why it is needed? High dose cancer treatment destroy the cancer cells and normal bone marrow cells also, because bone marrow cells they are also having rapidly dividing cells. So I discussed about this in the chemotherapy topic. So our natural bone marrow cells will be destroyed during this treatment. So we need to replace the healthy stem cells. So we have to do transplant. This transplant helps restart blood cell production. So here there are two different types of transplant: autologous, allogenic. Autologous means patient's own stem cells will be given to the patient. Allogenic means stem cells from the matched donor will be given. So here this is the major benefit. So it allows use of a very high dose therapy and improves survival. So here in this picture you can see patients receive pre-treatment to relieve stem cells from bone marrow into bloodstream. So here they are collecting the patient's own blood stem cells. Stem cells are separated and removed. Remaining blood is returned to patient. Stem cells are frozen until needed. It will be stored. Patient's own stem cells will be stored for some time until needed. So whenever it is needed, we need to thaw the stem cells and infused back into the patient. So this is the supportive treatment. Patient receives supportive treatment to help bone marrow regrow. So this is a more advanced therapy, gene therapy. So in this therapy, here we are modifying or replacing the faulty genes, which is responsible for the cancer production. So here replaces a defective gene with a healthy one. So if any gene is defective, for example, DNA repair gene, if it is defective, sometimes it may cause cancer. So at that condition, we have to replace the defective gene with a healthy one. This is the first condition. In the second case, we have to inactivate a harmful gene. For example, any due to any mutation, if a particular gene is mutated and it became very harmful and can able to induce the cancer, what we have to do? We have to inactivate the harmful gene, right? So that is the second case. In the third condition, we have to introduce a new gene to help fight a disease. Right. For example, as I said already, if DNA repair gene is inactivated, is if it is not working, in that condition, it is causing cancer. What we have to do, we have to introduce a new gene which can able to repair the DNA, right? So these are the in these three conditions we can use a gene therapy method. So here they are using vectors to deliver the genes. In the reproductive technology, you can know about these vectors and transgene process in very detailed manner. So these are the examples. CAR T therapy. Here, what they are doing, they modified T cells. Okay, I'll explain with the picture. So what are the advantages? It is highly targeted therapy. It reduces damage to the normal cells. But what are the limitation? It is highly expensive and technically it is very complex. So here you can see um, remove blood from patient to get the T cells. So we are removing the blood and we are collecting the T cells. So T cells are separated and removed. Okay. Then again blood will be given to the patients. So the removed T cells are genetically altered. Um, these T cells will get some special receptors called chimeric antigenic receptors. So these T cells are called CAR T cells. CAR means chimeric antigen receptor. So these chimeric antigen receptors will be multiplied by using cell culture method. Okay, millions of CAR T cells are grown. After that, what will happen? This um, cells will be injected to the patients. New CAR T cells introduced into the bloodstream. Right. So first, as I said, T cells are taken from the patient blood. So these CAR T cells, they have uh, very specialized cells. They can able to kill the cancer cells. So these T cells are taken from patient's blood. In the lab, they will be genetically modified. They will add a special receptors called CAR. This CAR, what it will do, it specifically recognize the cancer cells. Okay. So modified T cells are multiplied and put back to the patient. They find and destroy the cancer cells because they have some specific receptor to identify the cancer cells. So if we use the patient's own cells, here we can use the patient's own cells and very targeted, powerful against some blood cancers. Example, leukemia, it is very powerful against leukemia and lymphoma. So these are the major types of cancer therapy. So we have seen all the therapies in a detailed manner. I hope you understood everything. Thank you for listening.